Thank you. Good day, everyone. You're welcome once more to our monthly webinar for the GWIS International Chapter. And for today, for the September edition, we will have one of our members, Dr. Rishika Agarwal, who is a postdoctoral scientist in the University of Basel, Switzerland. So she will be talking to us today on the topic or title, Human Epidermal Organoids and their application in modeling epidermal diseases. So Dr. Rishika is also currently our public relations officer for the chapter and has been with us since 2022. So Rishika, you have the floor.
Thank you, Imelda. I'll start my screen share. Okay, can everybody see my screen?
Yes, please. I can see your screen.
Okay. So thank you for inviting me for sharing my research with everyone here. As Imelda mentioned, I'm a postdoctoral scientist at the University of Basel. And what I'll be talking about today is human epidermal organoids and how we can use it to model epidermal diseases. And I'll be using Darier's disease as an example. So my presentation will cover what the human skin is, what the epidermis is, what organoids are, the methods that I used to develop these organoids, and then how I characterize them. And then in the second part, we'll move to how I'm using the organoids to model Darie disease. So first of all, human skin is our largest organ. And while it's how the world sees us, it also has a lot of important functions. So it protects us from heat and cold, from microbes, from physical and chemical harm. And it's also involved in maintaining the water homeostasis of the body. The skin itself is made up of two main layers, which is the epidermis and the dermis, and then also has several appendages such as hair, sebaceous gland, the hair follicle, as well as the sweat glands. So to describe the epidermis in more detail, the epidermis starts at this, can everyone see my pointer?
Yes, I can see it.
Okay, perfect. So the epidermis starts at this basal layer and expresses these certain markers, so keratin 5, keratin 14, which are markers for the basal layer. And as these cells divide, these cells start to move upwards and differentiate. And so as they differentiate, they start forming the stratum spinosum, the stratum granulosum, and then ultimately the stratum corneum. So the stratum corneum is what is on the surface of the skin that we can see with our eyes. So currently the way to study the epidermis is usually using animal models or two-dimensional models where we just culture the keratinocytes in petri dishes or T75-25 flasks. There are also air-liquid interface models where it kind of mimics the in vivo system. So half of the tissue is in the culture media, whereas half is exposed to the air. And then a fairly new method to study the epidermis are organoids. So essentially, what are organoids? So organoid means resembling an organ. And so it's a collection of organ-specific cell type that develop from stem cells or organ progenitors and organize themselves through cell division and spatially arrange themselves such that they resemble how the epidermis looks in vivo. Now, how do we grow these organoids? So we take a skin biopsy from a healthy donor, and then we separate the epidermis using enzymatic dissociation, and then further isolate single cells using trypsin. These cells are then expanded until we have a certain number of cells. And then these are embedded in matrigel. Matrigel is the extracellular matrix. And it's basically a jelly-like substance that gives the cells a 3D space to grow compared to a 2D culture where they are growing only on the flat surface of the dish that we culture them in. We treat them with different growth factors and medias. I'll talk about it in the next slide. And in 7 days, these cells essentially grow into these organoids, which can then be collected from this matrigel and then analyzed for further analysis. So part of my PhD project was actually testing a lot of different media conditions that would allow the growth of these organoids. So already in the literature, this M1 existed. However, when I tested it, it actually did not give me good organoids. So you can see here that they are very sparse and very small. And so I tested a lot of different growth factors and eventually found that media 4, which contains specifically noggin and R-spondin, which are responsible for, which have a role in the Wnt pathway, are really essential for formation of the organoids. And already in this image, you can see that these organoids are large and well-formed. However, just looking at this structure with H&E and bright field imaging does not classify it as an organoid because it can just be a cluster of cells or a spheroid. And so for a cluster of cells to be classified as an organoid, it should fulfill 3 criteria. So it should contain the organ-specific cell types. These cells should be grouped together spatially, and then the organoid should be capable of replicating a function of the organ. And so to fulfill these criteria, I performed, so to show that the cells were specific to the epidermis, I performed immunohistochemistry and immunofluorescence stainings. So here in this upper panel, you see is what human skin looks like. And so you see that this marker for Ki67, keratin 14, and collagen 17 is expressed in this lowermost layer of the epidermis, which is the basal layer. And correspondingly, in the organoids, you notice that these markers are expressed on the periphery of the organoid. Whereas the differentiation markers, such as keratin 10, which is expressed in one layer above the basal layer, and filaggrin, which is expressed in the uppermost layer, start to also show their expression in the center of the organoid. So it's interesting to note that the organoid is kind of inside out. So this basal layer is on the outside, whereas the stratum corneum is towards the center. So here we see that this Ki67, the basal layer marker, is here, whereas in the organoids, it's on the outside, and the filaggrin, which is part of the stratum corneum, is towards the core of the organoid. So immunohistochemistry confirmed this, and then also immunofluorescence confirmed this. So I have here keratin 14 in white, which you see as this outline of the organoid, and then keratin 10, which is in the middle layer, and then filaggrin, which you can see in the core of the organoid. Now, interestingly, also, I was able to generate these organoids from primary keratinocytes, foreskin keratinocytes, and also Kurt cells. So Kurt cells are immortalized keratinocytes. So, primary cells we can keep in culture only up to 4 or 5 passages, but these immortalized cells can be kept in passage for a very long time. And so in the lab, they're a useful tool to have. Now, these organoids can be kept in culture for about a month. However, usually we keep them in culture up to day 14, because after day 14, they kind of stop growing in size. And then the longer I keep them in culture, the more the organoids become cornified, which is essentially that they lose their nuclei, and they are essentially only just the stratum corneum then, which is your dead cells. We also looked into transcriptomics. And so what we see is that as the organoids progress from day 0 to day 7, they cluster differently, indicating that there are different groups of genes that are upregulated as the cells undergo from a 2-dimensional structure to a 3-dimensional structure. And then we see that all of these pathways involved in the cornified envelope, keratinization, keratin filament, the desmosome formation. The desmosomes are the connections between the 2 keratinocytes of the epidermis. So these are all upregulated, indicating that this model is really useful to study the epidermis compared to a 2D model.